guys, welcome to my YouTube channel. I'm Dr. Shuchita Verma and today we are going to study everything about biotechnology. So we are going to start the unit of biotechnology for class 12, and this unit has two major chapters. The first chapter is principles and processes of biotechnology, and the second chapter is applications of biotechnology. So in this video tutorial we are going to discuss about various principles and processes in biotechnology. So let's get started. Biotechnology. So this is a combination of two words, it is bio and technology. So bio basically refers to living organisms and technology is the techniques involved in this area. Yeah. So biotechnology, as we go by the definition says that it is the use of living organisms or their enzymes or their molecules to obtain products or develop processes. To obtain products or to develop processes for the human welfare. For the human welfare. Yeah. So by definition, it is all the technology that is involved in the use of living organisms, how we can use different living organisms, including the microorganisms, how we can utilize their various enzymes that they produce, or maybe various other molecules that they produce for the benefit of human society, for the benefit of human welfare. And this includes products like insulin, various antibiotics, various vaccines, as well as various processes that are able to help somewhere to the society, that are able to contribute to the human welfare. Yeah. So before moving further, let me quickly summarize how far we have come in this area of biotechnology. Now, biotechnology is an advanced science, but then there are a number of applications that are available worldwide with the help of this technology. Yeah. Number one application is the use of insulin. Now, as we all know that diabetes has been declared as an pandemic by WHO. Yeah. So diabetes, what happens in the situation of diabetes, that our beta cells of islets of Langerhans, they are unable to produce the insulin because the beta cells basically get destroyed. So insulin is a hormone which is secreted by our body. And what is the function of insulin? Basically, insulin helps to regulate the concentration, the amount of sugar, blood sugar. So every time there is excess of there is excess of sugar in our blood, the insulin is released, and that insulin helps in the regulation of this sugar. But what happens in case of diabetes, that the patient is unable to synthesize this insulin in the right amount. So now diabetes is a big problem in so many countries these days. Even the children are suffering from type 1 diabetes and the adults are suffering from type 2 diabetes, where there is a resistance towards the insulin. So what scientists have done? Scientists have come up with a beautiful solution to it. Now, what they did is that they took the human insulin gene from the humans, and then they have inserted this gene into a prokaryote. And that prokaryote is E. coli. So now they have inserted this gene for the production of human insulin into the E. coli genome, and there this gene is being transcribed first and then translated to produce the desired insulin. Now, what industries do? They simply isolate this human insulin from the E. coli and then package it into the vials, and that is being marketed into the market, yeah, and that is being supplied into the market, and from where the patients can buy that insulin and can be given this insulin therapy. Now most of the diabetic patients, they take this insulin with the help of injections or maybe the insulin pens available into the market. So this is one very important and very successful application of biotechnology, yeah. So the first example, the first application that we have discussed about biotechnology is the insulin that is being produced in the E. coli. Yeah. Now the second application of biotechnology is the production of vaccines. Now I'll be taking the example of polio vaccine. Now polio vaccine, what happens is that we get the weakened or the killed version of the virus in case of a polio vaccine. So now how much quantity does a child need of for the polio vaccine to be immunized? Yeah, 2 drops of polio vaccine is being used for the proper immunization. Yeah, so whenever we are taking the vaccine, we are assuming that yes, we are being completely immunized. But in some cases, although the chances are very, very less, but still there is a risk factor associated with this kind of a vaccine, that even if by mistake the vaccine contains the live version of the virus, or maybe the virus is not being properly weakened, then the virus might cause an infection of polio into the administered individual, yeah, into the baby. So to avoid that, what we have done is that scientists have taken the protein present in the capsid of the virus. So now this protein is a product, yeah, and for every protein we need a gene, yeah, because it is the gene that is responsible for the production of a protein. Now what happens that because this protein is coming from the gene is first transcribed and then it is being translated into the protein. So now basically what scientists have done, that they have inserted this gene into the plant. Now the gene, when inserted into the plant, is now being transcribed and translated to produce this specific protein in the plant. One such example is banana. Yeah, what happens that the scientists, they insert the gene into the banana, and now that banana plant is producing the particular protein that is being responsible to elicit the antibody production in the patient. So these kind of vaccines are called as edible vaccines. Now when this banana is being given to a baby and the baby eats, then what happens? This protein is considered as an antigen, and this antigen is now responsible to induce the antibody response into the baby. So this way we can say that now the baby is completely immunized, as well as there is no risk factor associated with this type of animal vaccine. Why? Because we are not giving any killed version of the virus, nor we are giving any weakened version of the virus. So this is how, just by inserting a gene responsible for the production of the protein, wherein this protein will act as an antigen to the body, and then the body will start producing the antibodies against this particular protein or the antigen. So this is how we can immunize children, as well as we can be sure of the fact that there is no risk factor associated, as there is a little risk associated as compared to the weakened or the killed version of the viruses. Yeah, I've discussed 2 major applications of biotechnology. Another very beautiful wonderful application of biotechnology is the use of uh, you know protein that is produced in the firefly. So all of us know that fireflies, they glow in the dark, yeah. So basically, why does this firefly glows? The reason is a simple protein that is produced in it, and that protein is luciferin protein. So this is a protein that is being produced in this, and it is actually responsible for the glowing effect of these fireflies in the dark. So now what scientists have done, so before understanding this process, let us see what luciferin does. Yeah, so luciferin, in the presence of light, when the photons are available, it absorbs these photons. Okay, and when there is dark, means there is no light produced, no light available, then in that situation, this protein, it emits light, it emits some wavelengths. So basically, due to these wavelengths, the glowworm or the fireflies, it glows. So now what scientists have done, they have isolated the gene responsible for the production of this protein called luciferin. And then this gene is being introduced into the plants, and once inserted into the plant, the gene transcribes and then translates to produce this particular protein called luciferin. And then because of this, they have planted these plants along the roadsides, along the streets. So what happens during the day, that these plants, because of the presence of proteins, they absorb the light, and during the night when it is dark, the plants glow automatically. So there is no need for the external source of light, which actually saves a lot of energy. And then because of this particular mechanism, there is so much light even in the dark on the road. Now we don't need any external source of light, because of this biotechnological application of having this gene inserted into the plants, and then we'll have all the glow in the dark effect because of this particular protein luciferin. So we don't need any external lighting system for our street lights to for the light to be there. And yes, these are some applications of biotechnology, and there are millions more, thousands of more applications we're going to discuss in this chapter. And then this is all about, a little introduction to how beautifully this biotechnology is working to make our lives easier, better, and more comfortable. And now let us look at some of the historical aspects of this biotechnology. Let's go ahead. So, guys, when we talk about the historical developments for the biotechnology, the number one thing that comes to my mind is the rDNA technology, the rDNA molecule, the first rDNA molecule that was being produced. The scientists were Boyer and Cohen, and it was done in the year 1972 when the first rDNA molecule was being synthesized. So, what scientists did, they isolated the antibiotic resistance gene. So, they isolated the antibiotic resistance gene from the organism that is Salmonella typhi. Salmonella typhi. Alright. And this is the gene that they isolated, and they inserted this gene into the E. coli plasmid, the plasmid of E. coli. So we'll be discussing this process into the details in our next video, but this is just to give you a general idea how it happened and what it actually was about. So this is the Salmonella typhi antibiotic resistance gene, and this gene is being inserted into the plasmid of the E. coli. So it somehow looked like this. this is a part of the plasmid, and then into the plasmid, here the antibiotic gene was inserted. Yeah. So this is the first rDNA molecule, the recombinant DNA molecule that was being synthesized. Now there are a number of processes and other principles that I'll be discussing in my next video tutorials. If you have any query, just leave a comment in the comment section and just let me know how the video was, what are the things, what are the topics that you want to discuss in the video, and just leave a comment in the comment section, like the video, share and subscribe with your friends, and we are going to discuss more things about biotechnology, everything that is a part of this curriculum, this chapter, and a lot more. What is going on into the research? Where are the and what are the advances in the biotechnological research these days? Thank you so much. Goodbye.